Hello, my name is Joshua Mugabi. I welcome you to Coordination. We are going to look at learning outcome A, examine the role of plant hormones in tropism, photoperiodism, and the application of these processes in agricultural practices. We are going to look at the learning activity C and D. Learners carry out scientific investigation on the effect of ethylene on raw fruits. Learner writes a report and present their findings. And learners search for or visit a floriculture farm to find out the effect of the change in day length on plant flowering and application of changes in day length in floriculture. Item 1. Your school receives unripe green bananas for the boarding section. The matron complains that they take too long to ripen naturally. As a biology student, you suggest using ethylene, a natural plant hormone that speeds up fruit ripening. Task: design an investigation to find out how exposure to ethylene gas affects the ripening of raw bananas. The aim of our investigation: investigate how exposure to ethylene gas affects the ripening of raw bananas. This one will take us to our hypothesis. Bananas exposed to ethylene will ripen faster than those not exposed. Our variables: we have the independent variables that will be the presence of ethylene gas. Dependent variable will be the rate of ripening, color change, softness, and sugar content. Controlled variables that will be temperature, types of fruit, number of fruits, humidity, and container type. Experimental setup. Group A: bananas sealed in a paper bag with a ripe apple as a source of ethylene. Group B: bananas sealed in a bag without ethylene source. Observe changes daily for 5 to 7 days. Data correction. Color scoring when they change green to yellow. Texture, firm to soft. Smell intensity, and daily photographs. Conclusion: ethylene exposed fruits ripen faster, confirming the hypothesis. Item 2. A farmer wants his tomatoes to reach the market while firm but ripen just before sale. He hears that traders use ethylene to synchronize ripening. Task: explain how ethylene can be applied commercially and discuss any advantage or risks. Place mature green tomatoes in ripening chamber. Introduce ethylene gas at controlled concentrations, 100 to 150 parts per million. Maintain ideal temperature between 20 and 24 degrees Celsius. Ripening becomes uniform, improving market value. Risks: over-ripening, softening, spoilage if concentrations too high. Item 3. A fruit exporter aims to keep avocado firm during long-distance transport. Students must advise which hormone interacts with ethylene to delay ripening. Task: explain how abscisic acid, ABA, and ethylene interact and propose a solution. Abscisic acid maintains fluid dormancy and slows metabolic activity. Low temperatures and controlled atmosphere can suppress ethylene action. Recommendation: transport fruits in low oxygen containers at low temperature to inhibit ethylene synthesis. Let's look at the investigation on the effect of ethylene on raw fruits. We are going to write a report on the effect of ethylene on raw fruits. Our title: the influence of ethylene gas on the ripening rate of raw bananas. The aim of the experiment: to investigate how ethylene gas affects the ripening process of raw fruits. Introduction. Ethylene is a naturally occurring gaseous plant hormone responsible for ripening climatelic fruits such as bananas, tomatoes, mangoes, and apples. It triggers several physiological changes, including conversion of starch to sugar, breakdown of chlorophyll, the greening pigment, softening of tissues, aroma production. Understanding ethylene's effect is important both ecologically and agriculturally. Hypothesis: raw fruits exposed to ethylene gas ripen faster than fruits kept without ethylene exposure. Materials: 10 raw green bananas, 2 brown paper bags or tight container, a ripe apple as ethylene source, labels, observation sheets, and camera, which is optional. Procedures. Divide bananas into 2 groups of equal size. Group A, which is experimental, put bananas in a paper bag with a ripe apple. Group B, a control experiment, put bananas in a separate bag without an ethylene source. Seal both bags. Place them in the same room under identical environmental conditions. Observe daily for 5 to 7 days and record. Peel color, softness, sweet aroma, any sign of decay. The results, our data table. Without the number of days, Group A with ethylene, Group B no ethylene. On the first day, Group A, they were green and hard, and Group B were green and hard. On the second day, Group A with ethylene, they became light green. Group B with no ethylene, they are still green. On the third day, Group with ethylene, they developed yellow patches and became softer. Group B without ethylene were still light green. On the fourth day, Group with ethylene, most of them were yellow and soft. Group B without ethylene, they were greenish yellow. On the fifth day, Group with ethylene, they were fully yellow and aromatic. Group B without ethylene, they were partially yellow. On the sixth day, the Group with ethylene had started to develop overripe patches, and Group B with no ethylene were yellow and still firm. Our observation, Group A ripened significantly faster than Group B. Analysis. Ethylene from the ripe apple initiated biochemical processes in Group A bananas, increased respiration, conversion of starch to sugar, breakdown of chlorophyll leading to yellow color, softening of tissues. Group B, lacking ethylene, ripened at a much slower natural pace. Our conclusion. The hypothesis is supported. Exposure to ethylene gas accelerates the ripening of raw fruits. This explains why fruits stored together tend to ripen simultaneously and why traders use ethylene commercially. Let us now look at the ecological and agricultural significance of ethylene. We shall start with ecological significance. Ethylene ensures seed dispersal by ripening fruits when seeds are mature. Ripening attracts animals, promoting ecosystem biodiversity through seed spreading. It coordinates fruit drops and leaf abscission in natural plant cycles. Agricultural significance of ethylene. Controlled ripening. Farmers can ripen fruits on demand for markets. Uniform ripening improves quality, grading, and consumer appeal. Reduced post-harvest loss. Synchronizing ripening reduces waste. Storage and transport strategies. Delayed ripening by preventing ethylene exposure. Use of ripening chambers for large-scale operations. Let's now look at commercial application of ethylene. Used in banana and tomato ripening chambers. Enables staggered harvesting and market supply control. Speeds up degreening of citrus fruits. Used to synchronize flowering in pineapple plantations. Helps remove unwanted dormancy in tubers like potato sprouting. Let's now look at the effect of change in day length on plant flowering. Item 1. Your school garden has flowering sunflowers that begin flowering earlier when the rain season starts, but delay flowering when the day length becomes shorter during cloud month. Task. Explain how changes in day length influence the flowering of these sunflowers. Explain how changes in the day length influences the flowering of these sunflowers. Sunflowers are long day plants. They flower when days are long and nights are short. During rain season, when days are brighter and longer, they get more light hours, stimulating early flowering. During cloud and shorter days, they remain vegetative for longer. The plant uses the phytochrome system to measure night length. Item 2. A farmer complains that her chrysanthemums do not produce flowers during months with long days. She wants to understand why. Task. Explain the photoperiodic reason for the delayed flowering. Chrysanthemums are short-day plants. They require long uninterrupted nights to trigger flowering. When days are long or nights are short, the flowering hormone floriculture is not produced. Therefore, flowering is delayed in seasons with long day light. Item 3. A greenhouse uses security light at night. The gardener note says that poinsettia fails to flower. Task. Describe how artificial light influences the flowering processes. Light at night interrupts the critical day dark period of short day plants, like poinsettias. The interruption resets the plant's internal photoperiodic clock. Phytochrome remains in the active form, preventing flowering. Results: the plants stay vegetative. Now let us look at the application of changes in day length in floriculture. Item 1. Commercial flower grower want rose ready for Valentine's Day. Natural day length at that time is short. They consider using artificial lighting. Task. Explain how altering day length can ensure rose flower in time for market. Roses are long day plants or day neutral species depending on the variety. For long days varieties, farmers extend day length using supplementary lights. Artificial lighting increases photoperiod and stimulates flowering even during short natural days. This allows precise scheduling of flower production for markets. Item 2. A farmer wants poinsettia flowering exactly in December for Christmas sales. However, natural day length may not give consistent results. Task. Describe how day length manipulation can help synchronize flowering. Poinsettia are short day plants. Floriculturists use black cloth shading to create long nights. They cover plants in the afternoon to ensure at least 12 to 14 hours of darkness. This triggers flowering at the exact time needed for holiday sales. Removing light at night ensures production of floriculture. Item 3. A greenhouse operator wants carnations and lilies, mostly long day plants, to flower continuously. Explain how manipulating photoperiod can make year-round flower production possible. Use extended daylight, incandescent or LED lights to increase long days when during winter. Turn lights on from 10 p.m. to 2 a.m. Night break lighting to interrupt the long night. The night break technique tricks plants into thinking days are longer. As a result, flowering continues throughout the year. This stabilizes supply and maximizes profits. Item 4. An export farmer wants to improve uniformity and size of cut flowers. They are advised to control both day length and light intensity. Describe how photoperiod control improves flower quality. Long day treatments improve stem elongation, increase vase length. Manipulating day length ensures uniform flowering, improving grading standards. Short day treatments can enhance color development in some species. Combining photoperiod control with temperature regulation leads to premium quality blooms to or for export. Thank you very much for participating in coordination. Learning outcome A, examine the role of plant hormones in tropism, photoperiodism, and application of these processes in agricultural practices. We have looked at learning activity C and D. Learners carry out scientific investigation on the effect of ethylene on raw fruits, and the learners write a report and present their findings. Learners search for or visit a floriculture farm to find out the effect of change in day length and plant flowering, and the application of changes in the day length in floriculture. If this activity has been very useful to you, please visit me at Mugabi Joshua Biology on YouTube.